welcome everybody who's joined us virtually to this latest installment of our virtual Water Ambassadors program. This is our stormwater education program for Martin County, and also want to show you on the screen on your that I'm showing you on your right are the lists of our scheduled dates. And as Lisa had mentioned, I'm here with Lisa Krimsky. We're both from UF and Florida Sea Grant. If you have any other topics you're interested in, please fill that out in the chat. And the chat is what you'll use to enter any questions. So we are scheduled. You can take a look at that. I think we'll also put the links, the registration links in the chat as well, so you can register for them if you haven't already. So just to do a little housekeeping here. Next slide. Yeah, so if you're not familiar with Zoom, we just want to remind you your microphone is muted. And if you can't hear us, click on that little arrow or caret symbol, and then that can adjust your audio settings. It is for some people who have maybe a less strong internet connections, it does help to turn off your video during the presentation and kind of help solve some of the issues with bandwidth. That's something that we typically do. And you can see who's on by clicking on the participants list. Also, again, as I mentioned, if you have any questions, you can ask any questions anytime. You enter it in the chat, and we'll read through them at the end of the presentations. It's a little bit easier to manage that way. And of course, we don't want you to leave, but if you need to leave, you can click that leave meeting button there. And if the view settings will help you adjust what the screen size of the slides. You can move the participant window around a little bit just to help with that as well. Okay, this webinar is being recorded, and we'll make that available online on the web address that you see here. This is the University of Florida Martin County Extension website under the Sea Grant tab, and you can view this recording later and other recordings from last year and earlier this year on this website. And typically we can get these recordings posted within a couple days, usually maybe 3 to 4 days at the most. And again, as I mentioned, please use the chat feature to ask your questions, and we will get to your questions at the end of the presentation. And so with that, I'd like to introduce Dr. Malcom MacFarland from Florida Atlantic University, specifically at the Harbor Branch Oceanographic Institute up in Fort Pierce. Dr. MacFarland has been involved in monitoring phytoplankton in both the St. Lucie Estuary and the Indian River Lagoon for the last several years, particularly with an eye towards harmful algal blooms. Harmful algal blooms are a big concern in the Indian River Lagoon. There's several blooms going on right now, and if you recall from 2016 and 2018, we had some pretty bad cyanobacteria or blue-green algae blooms in the St. Lucie Estuary. So I think he'll talk a bit about all of that work. And with that, I'll let Dr. MacFarland take it away. I'll go ahead and stop sharing here.
Thank you very much, Vincent and Lisa. Thanks for having me. Everyone can hear me okay?
Yes, you sound great.
I guess I'll go ahead and start sharing my screen. Okay. And you guys can see my screen now?
We can.
All right, fantastic. So as Vincent said, I'm basically a phytoplankton ecologist. I study microscopic algae, and I am mostly coming from a marine background. So my training is in biological oceanography, really. But I have been studying a lot of the organisms that are responsible for harmful algal blooms for a while, especially since I've come down to Florida, where harmful algal blooms seem to be a real issue, more so than maybe anywhere else in the world that I've been working. So this is really fascinating area of ecological research for me, and it's really important on both a local and a national level. So I'm going to talk to you a little bit about the work I've been doing locally, and some of the things we found, and some of the methods that we use. So to start off, so we're all on the same page here, what are harmful algal blooms, or HABs? Basically, these are very high concentrations of microscopic algae. Microscopic algae, also called phytoplankton. And these actually include a really wide, diverse array of different kinds of organisms. They're generally very small and microscopic, so you can't usually see the individual cells with your naked eye, but they can be extremely abundant in natural water bodies, both freshwater and saltwater. So some of the types that we see that are responsible for harmful blooms are things called dinoflagellates, diatoms, cyanobacteria, which are actually prokaryotic organisms, and then various other strange and not often very common types of organisms called pelagophytes or rhipidophytes and promesiophytes. These are all extremely diverse in a taxonomic or an evolutionary sense. They're really quite different organisms, even though they're all kind of lumped together under what we call microscopic algae. So some of the images you're seeing here are just some examples. On the far left, this is a type of dinoflagellate called Alexandrium, and what you're seeing here is 4 cells in a chain. To the right of that, you're seeing a cyanobacteria. This is Microcystis aeruginosa, and this is the one that has caused a big problem within recent years in Lake Okeechobee and in the St. Lucie Estuary. And this is a whole bunch of cells viewed under epifluorescence microscopy. So all these red dots, these are all small individual cells you see in this larger colony. And to the right of that is a dinophysis, another type of dinoflagellate. And to the right of that is a scanning electron micrograph of another type of dinoflagellate called Pyridinium bahamense. So you can see that these are, there's lots of, they're all single-celled organisms, but they have lots of characteristic morphological features that differentiate them from each other. They vary a lot in size and shape and how they grow, and some of the small features like whether they have these pores in their cells and these different types of structures on their cells. So there's a big diverse array of these things. And whenever I talk about these guys, I want to make sure people understand that they're not all evil and bad and harmful. They are actually, phytoplankton in general and these microscopic algae are really important components of marine ecosystems and freshwater ecosystems. They basically form the base of the food chain, and they are absolutely critical. Without these types of organisms, we wouldn't have zooplankton, we wouldn't have fish, and all these other higher trophic levels that are supported by these small microscopic plants, basically. So they're plants. They, ecologically speaking, they're plants. They fulfill that same role. They take in light from the sun and nutrients, and they do photosynthesis, and they provide us with oxygen, and they provide food for herbivores at the base of the food chain. So in this slide, what you see in green here is sort of the distribution of these microscopic algae across the globe. And they're really quite abundant. At a global level, they're extremely important. They do about as much photosynthesis as all the land plants do. So they're really responsible for about half of the oxygen that's in the atmosphere that we breathe. So these are really critical, essential components of ecosystems on Earth. They are some of the species, like these diatoms in the bottom left, are incredibly ornate, and the structures that they create at a nano scale are really quite intricate and pretty amazing. It's something that has really fascinated biologists and ecologists for ever since the microscope was invented, basically, and we discovered that these types of organisms exist. And in fact, people have been so fascinated with them, they've turned a lot of these organisms into art. And what you see in the middle here is two arranged slides consisting of different shells of different types of phytoplankton. These are all diatoms. And what someone has very painstakingly done is arrange these into nice geometric patterns. And this was sort of a famous hobby of early naturalists in the late 1800s to do something like this. And on the right, you see that these are some illustrations, some old illustrations by a scientist and early naturalist named Ernst Haeckel. And he was absolutely fascinated by these types of organisms. And you can see he's drawn, maybe in a little bit of an exaggerated way, but not too exaggerated, all the different types of organisms that he was able to discover. So again, I hope if you come away with this, with anything from this talk, it's that these are really a fascinating group of organisms that are not just solely responsible for harmful algal blooms. They are really essential part of our ecosystems. So I mentioned that phytoplankton are basically plants. So how do they grow? So, well, they need basically three things, like any plant does. They need light, they need nutrients, and they need temperature, or a certain temperature range. So obviously they do photosynthesis, so they need light. Nutrients are basically the same types of things that would be in fertilizer that you put on your lawn, things like nitrogen, phosphorus, various trace metals. And they need these to fuel their growth. And in order to get large blooms of these organisms, you need lots of nutrients. Temperature, generally warmer temperatures lead to higher growth, but this can be a little bit species dependent. So some species are adapted to cold temperatures, and some are adapted to warmer temperatures. So some diatoms that we find in the Indian River Lagoon, for example, can bloom this time of year when the temperatures are relatively cold, whereas other types of organisms can only bloom in the summertime. So it's important to note that 2 out of these 3 requirements are really right now being heavily impacted by human activity, both locally and globally. So temperature in particular is slowly going up due to global warming. But on a global level, and this is even slight changes in temperature on the order of a degree can be important here. And nutrients locally, a lot of places, and Florida in particular, has a problem with the nutrients that people are introducing into fresh waters that actually ends up in our coastal ecosystems. Things like nitrogen and phosphorus runoff are a real problem. These come from wastewater treatment facilities in urban areas, septic systems, and also agricultural practices that are adding fertilizer to grow crops. And a lot of that nutrients runs off into freshwater streams, canals, ends up in St. Lucie Estuary or in the Indian River Lagoon, and that is a problem for fueling some of these blooms. So what makes these things harmful? There are actually a variety of ways where these high concentrations of phytoplankton can be harmful. Some of them produce toxic compounds. And obviously this has a direct impact on anyone who eats these organisms or who perhaps eats fish that has fed on these organisms. But they can also lead to anoxia in the environment. And anoxia is basically a lack of oxygen. When you get really high biomass from these blooms, the organisms themselves actually need to consume oxygen when they're not doing photosynthesis, like at night. And when there's really high concentrations, they can consume a lot of that oxygen. And then when they die, the biomass fuels the growth of lots of bacteria. And the bacteria can consume a ton of oxygen as well. And so this can end up with waters with very low oxygen content, and that can result in fish not having enough oxygen to breathe, and it can lead to large fish kills. Another way that they can be harmful, especially in the Indian River Lagoon, is through shading. So when there's very high concentrations of cells in the water, light cannot penetrate to the bottom. And that means things like the natural seagrass beds, which are important pieces of the ecosystem in the Indian River Lagoon, can't get enough light to grow. And so harmful algal blooms can kill a lot of seagrass. And we've seen that happening in the Indian River Lagoon. Some other unusual ways that they can be problematic is through things like mucus secretions. So some of these organisms, particularly this one in the middle here, this Akashiwa sanguinea, it's been associated with the production of lots of extracellular compounds, basically goo that it extrudes from its cells. And in places on the west coast of the United States, in California, it's thought that this goo actually gets on the feathers of seabirds. It can destroy their waterproofing, and then the seabirds can die from hypothermia. And of course, there can also be economic impacts from a lot of these blooms. That's another way they can be harmful to humans. A lot of them are unsightly. They cause a mess. They cause fish kills. It's not really good for tourism, and it's definitely not good for fishing. And so that can impact local economies pretty badly. So why should we care? Well, as I said, algal toxins are a direct threat to animal and human health. On a broader sense, they can really pose a risk to the ecosystem stability, its structure, and its function. So when we have high concentrations of these phytoplankton, things get out of balance, and the normal relationships between organisms in the ecosystem is disrupted. And that can cause the whole system to sort of collapse or reset in certain ways that are not beneficial to most of the organisms that usually live there. Of course, it can be detrimental to tourism and local economies, but the problem also seems to be increasing over time. As I said, we're impacting things like temperature and nutrients in coastal water bodies, and this seems to be leading on a local, on a national, and even on a global level to an increase in these harmful algal blooms. And we need to really understand how these factors are impacting blooms so we can figure out the best strategies to employ to minimize the blooms themselves and their impacts. So you can see on this slide, I've included a bunch of recent local headlines. Well, these are a couple of years old now, but these are things that have happened locally. This is causing some real problems in different parts of the lagoon. You can see on the bottom here, this is a fish kill that happened in the northern Indian River Lagoon as a result of a harmful algal bloom that consumed all the oxygen in the water, and all these fish trapped in the canal basically died. We can see along the coastline other organisms like the Florida red tide have resulted in lots of dead fish piling up on beaches. And this is more common on the west coast of Florida, but has and can come over to our east coast and impact the coastal areas around us. So as I said, harmful algal blooms seem to be increasing. Nationally, this shows the difference between 1972, so a while ago now, 50 years ago, there were a handful of sightings around the coastal US. It wasn't unheard of, but it wasn't terribly common. Now, and this is even a few years old at this point, this plot, so I would expect it to be even worse. Now we see that these things are popping up all over the place. Basically, the entire coastline has a problem. The entire coastline of the US can have a problem with these harmful algal blooms. And it's all different species, different types of problems associated with these different species, but they can all be caused by similar factors, and we're seeing an increase in those factors leading to an increase in blooms. And Florida in particular, you can see down here, is basically covered with all different types of harmful algal blooms. So we've got it bad where we are, unfortunately. So zooming in on Florida, this is a map that shows some of the major blooms that we've been experiencing over the past few years in different parts of Florida. So these on the right here, I've listed, these are some of the major species or genera, types of algae that we've been seeing causing problematic blooms, and the ones that we've really been on the lookout for when we're monitoring for these things. So up in the northern IRL in particular, we've got a lot going on. There's been a persistent bloom of an organism called Oreoumbra lagunensis, and that has been going on for years actually, and it sort of moves around a bit in the upper lagoon, but it's almost always there. Very high cell concentrations of this particular type of organism. Thankfully, it does not seem to be toxic, but its high concentrations can lead to anoxia, and also have been shading out seagrasses in the area. Also up there, we have annual blooms. They occur almost every summer of an organism called Pyridinium bahamense. And this is a dinoflagellate. It produces a toxin called saxitoxin, which is a neurological toxin that actually causes what's called paralytic shellfish poisoning. So if you consume this, and if other animals consume this, it can be fatal in high enough concentrations. Throughout much of the lagoon, on the east coast here, we see a type of diatom called Pseudonychia, and there are a variety of species that we see in this genus, and these produce a toxin called domoic acid. And I'll talk more a little about that later, I think. Then in the central part of Florida's freshwaters, Lake Okeechobee, and then in the canals and rivers that transport Lake Okeechobee water to the coast, we see Microcystis aeruginosa, which is a cyanobacteria, primarily freshwater, but it's getting introduced into our coastal ecosystems through runoff. On the west coast, we have Karenia brevis. Primarily on the west coast, we have Karenia brevis, which is another type of dinoflagellate. It blooms almost annually. Probably everyone's heard of this in Florida. Produces lots of fish kills. It produces a toxin that is very toxic to fish, and in even moderate concentrations can cause massive fish kills. It's also an irritant to people who breathe it in. And usually that's on the west coast, but sometimes it can come around to the east coast. Down in the southern parts of Florida, around the Keys, we see high concentrations of cyanobacteria. We also see some of these high concentrations of cyanobacteria in the lagoon. These can be very tiny, not necessarily toxic, but still can cause problems. And then we also see something called Gambier discus, which is not quite as common, but that can cause, that produces ciguataria, which is a toxin as well. So I'll go through a few of these more important organisms in more detail here. so you get a better understanding of what some of these critters are and why they're potentially a problem, and why they're blooming. And it's all different depending on the different species. So first, Microcystis aeruginosa. This is the one that caused all the green slime in the St. Lucie Estuary back in 2016 and 2018. This is a cyanobacteria, as I've said. This is a picture on the right of it under a light microscope. And what you see here is lots of individual cells in this big colony. And the colonies are like sort of these big loose aggregations of cells. And they can get quite large. You can have colonies that are centimeters in size floating around. So you can definitely see this with your naked eye. This produces microcystin, which is a liver toxin. So consuming this organism and this toxin can cause liver failure and cancer. One of the unique things about this organism is that it has these gas vacuoles or vesicles within its cells. And in the middle here, you can see an electron micrograph of just a single cell, and all these honeycomb-like features that you see, these are the gas vesicles. And this allows this organism to float, which gives it a real advantage in places like Lake Okeechobee, where it really likes to bloom. Lake Okeechobee waters are very turbid, so light does not penetrate very deep into the water. So if this organism is mixed or is not at the surface, it can be very light limited. But because it has all these gas vesicles, it floats near the surface. And this allows it to grow like crazy, even when the water is very turbid and other organisms can't. So it blooms in the summer, in the summer months, pretty regularly on Lake Okeechobee. And when water levels get high during the rainy season, they have to release water to the coastal estuaries. And if the timing is right or wrong, and the microcystis is in the right or wrong place at the wrong time, it can be transported with that water down the St. Lucie Canal and river and into our coastal estuaries, where it can become trapped there to some extent and really cause a big mess. So this shows up pretty easily on satellite images. So this is an image of the lake you see on the left here, and all this sort of green that's through the center of the lake here, that is the microcystis. And it shows up great on satellites because it floats right near the surface and it's bright green. So this is what it looks like on the right here. This was an image taken from the St. Lucie Estuary after it had been transported through the canal, and it kind of got trapped in areas like this and causes a big mess. And this can be really dangerous for people who don't understand that it's toxic. There were a couple cases, I think at least, of pets dying because dogs drank some of this water and ended up suffering liver failure as a result. So the next one I thought I'd talk about is this Oreoumbra lagunensis. This is another small cell organism. It does not form colonies. It's actually really tricky to identify because if you look at it under the light microscope, all you see is these little dots. Not much to go on. We know that it's what it is because of electron microscopy images like this one, and also from people doing genetic studies and looking at the genotyping of this organism. So this is a type of organism called a pelagophyte, which is kind of unusual. It's not a very common class of organism, and originally it was associated with the open ocean environment and deep open ocean environments. And it's thought that it sort of found a niche within these coastal ecosystems where it was really able to explode and take off. And this has happened in the northern Indian River Lagoon up by Titusville, Cocoa Beach area, Banana River Lagoon and Mosquito Lagoon. It's also happened in places in Texas, Laguna Madre. And the reason it's called Oreoumbra lagunensis is actually because of the persistent bloom where it was identified in Laguna Madre. So this bloom, one of the interesting things about this bloom is that it's very localized in the Indian River Lagoon. It seems to stick around up here in the northern Indian River Lagoon, and it's not really found anywhere else. So one of the big questions that I've had in studying the distribution of this organism is why does it not spread further south, which is kind of interesting. We don't know the answer to that yet, but that's one of the things that we're trying to figure out. So it's not toxic as far as we know, but it does lead to anoxia, which can cause fish kills and can cause seagrass loss. So this is one that we are keeping an eye on constantly, or as frequently as we can. And so while I am usually monitoring things down in the southern half of the Indian River Lagoon, we're always keeping an eye out for it to see how far south it'll spread. So these are just some of the images of what the water looks like, and it gives you a real sense of how it can cause this shading problem for seagrass. So here's an aerial photograph. You can see this brown water is really all the Oreoumbra lagunensis because of all the Oreoumbra lagunensis cells. And it's the reason we call it a brown tide. Closer up, you can see the water really does look, I mean, it's like milky coffee. And then this image here is the results of a fish kill caused by that organism. So the one that people probably have heard most of is this Karenia brevis. This is the Florida red tide. And it's really common on the west coast of Florida. It is a dinoflagellate, which is a common type of marine algae. But this one produces a toxin called brevetoxin. It's a neurotoxin. It attacks the neurological pathways of fishes, but it's also a respiratory irritant. So when there's strong blooms of this along the coastline, wave action and winds can sort of bring some of these compounds into the air, into the atmosphere. And when you breathe that in, it really causes a lot of irritation to your throat, your nose, and even your eyes, and it makes you cough. And people who are asthmatic or who have other respiratory problems are really at risk from some of this stuff. And as you can see in the bottom image here, it can really cause devastating fish kills. It's usually found in the ocean proper, so it's not really common to find it in the lagoons like the Indian River Lagoon, although I think you can get up into the Caloosahatchee River and Tampa Bay estuaries. So normally this is on the West Coast, but what can happen, and what did happen in, I believe it was 2018, the bloom develops along the west coast of Florida, and then the loop current and the Florida current in the Gulf Stream can sort of grab that bloom and carry it around the tip of the peninsula. And then sort of these recirculating currents that are associated with the Gulf Stream as it moves north, you can see some of these little eddies that spin off, they bring it over onto the coastline and let it hang there on the East Coast. And this is exactly what happened a couple of years ago. Really bad bloom on the West Coast, managed to spill over into the East Coast. And we saw fish kills along the Eastern coast, which even though they're normally restricted to the West Coast. And this seems to be about a once in every 10-year type of phenomenon. So it's not unheard of, but it's not terribly common. But as I said, we are impacting the frequency and the intensity of these blooms through climate change and nutrient runoff. And this may become more common. So we have to keep an eye on this and see how things develop over time. So another really interesting critter that grows up again in the northern IRL, but we've seen it throughout the Indian River Lagoon, but this is where we see most of the blooms. This is a dinoflagellate called Pyridinium bahamens, and this produces saxitoxin, as I said previously, which can cause paralytic shellfish poisoning. It's really quite toxic. Even small amounts of this can be deadly. Saxitoxin is actually listed by, I believe, the EPA or OSHA as an acutely toxic compound. And we have some cultures of this guy in our lab, but the environmental health and safety people are always nagging me about being careful with it because it's it's listed as something very toxic. Thankfully, we don't work with the pure compound, just the cells, so it's pretty safe. This is a motile organism, and when it blooms, it tends to form these aggregations that you see at the surface, these clouds of cells in the water, and locally it can become quite concentrated because it can swim. So you can see these streaks on the water sometimes, and it's a very rust red color. So this is another red tide type organism. These are some images of what it looks like under the light microscope and the scanning electron microscope. It blooms pretty much every summer to some degree in the northern IRL. Some summers are worse than others. This past one wasn't too bad, but it was still definitely there. And one really interesting thing about this is that it's very bioluminescent. So in this image, you can see the bioluminescence that's generated by this organism in the prop wash of a motorboat. And when it's disturbed in the water, when it's stirred up, basically it will flash. And at night, this can be a really impressive display. In fact, as you can see here, it's listed as something to put on your bucket list. And there are companies that are organizing kayak tours through these blooms at night. Unfortunately, probably most people don't realize that it's quite a toxic bloom. Now, it's probably not toxic and not going to be a problem to anyone unless they ingest it, but I wouldn't recommend going swimming in it, just on the off chance you get a mouthful. But it's responsible for a lot of the bioluminescence that's up there, and also potentially responsible for fish kills because of its toxins. All right, so one more. I think this is the last one I was gonna focus on here. This is a diatom called Pseudonychia, and it produces a toxic compound called domoic acid. Diatoms are not usually considered harmful algae, so this is kind of an interesting outlier. Diatoms are usually what we would consider good algae. They are really important members of the food chain and coastal ecosystems. But this guy produces domoic acid, which is another type of neurotoxin, and it actually can be responsible for memory loss and brain damage. So this one causes what's called amnesic shellfish poisoning, and people have actually suffered amnesia and memory loss due to ingesting this. It's a big problem on the West Coast as well, so not just here on the East Coast of Florida, but the coast of California, West Coast of the US. And there it's responsible for fish kills as well as problems with seabirds and sea lions. it's also thought that this could be the organism that inspired Alfred Hitchcock's The Birds, and a bloom of Pseudonychia could have been responsible for the erratic behavior of birds on the West Coast that inspired that movie. I don't know how true that is, but a little interesting factoid. So we're monitoring this one closely in the Indian River Lagoon. It blooms, unlike some of the others that bloom during the summer, this one can bloom during the winter, sort of this time of year. Diatoms are often well adapted to colder conditions. And so we see this blooming in the winter and spring when it does bloom. But unlike some of the others also, it's not always toxic. So this may or may not produce toxin depending on the species or depending on its particular physiology at the time. So not only are we interested in figuring out if this particular algae is there, but also what physiological conditions lead it to produce toxin. And this toxin is out there. We can measure it. We've measured it in the lagoon. And it's also been measured by some of my colleagues in fish and sea turtles. So it's getting into the food chain. So how much damage is it doing? What sort of problems is it causing, if any, is something that we're really interested in trying to figure out. So those are some of the most important ones that we've been studying in the Indian River Lagoon. And they're out there a lot, and their abundance is something that is really dynamic. It's always changing, and so it really necessitates continual monitoring to figure out who's there, how abundant are they toxic, and what is driving these different bloom dynamics that we're seeing. So there are also many other organisms that are out there that could potentially form harmful algal blooms, but we have not seen them do that yet necessarily in many cases. So this on the far left is, I showed this image way back in the beginning. This is another type of toxic dinoflagellate that produces a saxitoxin like the pyridinium, and we've seen this in the lagoon, but we've never seen it bloom. This Lingbia is a type of cyanobacteria that also forms toxins, but these guys form mats, big filamentous mats that sort of sink to the bottom. This Akashiwo sanguinea, this was the one that produces the excreted mucus. We've seen this in the St. Lucie Estuary, but we haven't seen major problem blooms yet, thankfully. This Dinophysis produces another type of toxin. We've seen that out there. We see it fairly regularly, but we've never seen very high concentrations. This Chaetoceros species is something we've seen along the coast. There are a lot of different species of these. I think I forgot to mention this earlier, but this can be harmful in a completely different way than a lot of these others. It doesn't produce a toxin, but if you can look carefully on these spines that it has, these spines are all made of glass. It makes a shell out of silica, and these glass spines even have smaller spines on them, like thorns on a rose bush. These can be very irritating to the gills of fish, and they can actually, in high concentrations, cause fish to suffocate and die because they get caught and irritate the gills of fish. So this is another type of organism that can be a problem. And we see it out there, but we haven't seen problems from it yet, thankfully. So what are we doing? Well, what we do to monitor this stuff and to try and figure out where and why it's blooming, what we do is primarily we go out and we collect samples. And this is some action shots of us going out on Harbor Branch boats, and we collect samples from various parts of the lagoon. While we're out there, we will collect some environmental water quality measurements as well. This is some of the in situ instrumentation that we use to do that. Occasionally we encounter very nasty blooms like this one here that we sample, but usually the water is more clear like this, thankfully. But in any case, we want to know who's there and how abundant they are. So we bring those samples back to the lab. We look at them under the microscope, and we might see something like this under the microscope. This is our trusty Nikon microscope that we use for that. We'll also use an instrument called a flow cytometer. And this is our flow cytometer that we have in the lab here. This produces data that looks like this down here. And what it does is it counts individual cells for us one at a time. And this is really good for counting the small cells like the Oreoumbra. So this plot, what you see on this plot here, every point represents an individual cell. And then we can categorize those cells based on how much chlorophyll they have or how big they are. And we can sort of group them into different types and count how abundant they are. So this is good for identifying certain organisms and counting them more efficiently than what you can do under the microscope. And then sometimes we also use an instrument which is sort of a combination of the microscope and a flow cytometer called a flow cam. This takes pictures of all the different types of suspended cells that are in the water as it pumps through, as it pumps a sample through a flow cell, sort of automating the microscopy process. So we end up with all this data and all these images that we can identify who is there, we can count who is there. And we're doing that pretty regularly from a number of sites in the estuary and in the lagoon, in particular these 5 sample sites here. And so for each of these sample sites, we have abundance of these different types of algae. And we get something that looks like this. So this is a plot from mostly from 2019. At each of these different sites, we have the number of cells that's in the water for all the cells. And then we also have data that's just, say, for this, as an example, just for this Pseudonychia that I was talking about. And you can see it varies a lot. There is a lot of variation in the abundance of these guys over time and from place to place. So it's a really dynamic system, and it's very challenging to keep track of who's there and how abundant they are, and are they going to be a problem? So this takes a lot of work, but we're doing the best we can. And the more people who are doing this, the better, because there's a lot to keep track of. This is an example of just some more lagoon-wide data that we have. This was collected from 2018. And this just shows the distribution of that Oreoumbra lagunensis. You can see there's a little bit of it around here, but mostly it's in the Banana River Lagoon where it was most concentrated. The circle size here indicates how much of it was in the water. So these bigger circles are showing that that's where there's lots of cells of this Oreoumbra. And then we also have, not me personally, but Harbor Branch maintains a network of observatory systems that are installed throughout the lagoon. And these things measure things like water temperature and salinity and chlorophyll. And these are measuring every hour, on the hour, 24/7, 365 days a year. So this is a huge asset that can really help us figure out what's going on in the lagoon, what is driving these blooms, and what sort of conditions are leading to bloom formation. All this data is publicly available. You can go check it out at erlon.org. So we have some of those sample sites that I was showing previously are designed to line up with these some of these in situ sensors so that we have really good in-water data that we can correlate with our cell count data to try and figure out what's going on. So we're measuring toxins directly as well as counting these cells. We're measuring saxitoxin, domoic acid, and microcystin using a technique called ELISA. And we can do this pretty much for every water sample we collect. We can count the cells. We can also measure the toxins. And this is an example of some of that data. So this microcystin, which is associated with the Microcystis aeruginosa, which comes from the lake, which I was telling you about, where you can see these big red circles down here. That's where we're seeing that toxin, which makes a lot of sense because that's where we're seeing the Microcystis. The saxitoxin, which comes from that pyridinium that I was talking about, we can see that up here in the Banana River. That's where we're finding that organism and that toxin, so that makes sense. But this domoic acid, which is the Pseudonychia, produced by the Pseudonychia, which I was talking about, we're finding that all over the place. Not high concentrations, but enough to be measured and really pretty much everywhere. We're also looking at how these toxins get into people. This is really interesting work that was done mostly by Adam Schaefer, a colleague of mine that I helped with. So on the left here, you're seeing microcystin concentrations in the environment through June to September in 2018. And around July, we saw really high concentrations of microcystin in the St. Lucie Estuary after it had been transported by the lake. And people, this is the concentration of that toxin in people's nasal passageways, which we measured using nasal swabs on volunteers. The concentrations of that toxin were relatively high when the concentrations of the toxin were high in the environment. And then as we sort of missed the beginning, but we caught the tail end of it. As the toxin went away, so did it go away in people's noses. So people, it is possible for people to be getting exposed to these toxins. Whether or not these are problematic levels, whether these are going to lead to illness, we don't know. Hopefully not, but it does seem to be detectable. Another thing we're doing is we're looking at metagenomics. So this is not what I'm doing directly, but working with some colleagues, we're looking for genes of these organisms so we can identify who's there based on just the DNA that's out there in a water sample. Also looking directly for genes of the toxins. And we can hopefully maybe look at how active those genes are as well. So that's really cool stuff. We're also using a variety of bio-optical and satellite remote sensing techniques to monitor stuff as well. So on the left here, you can see some of the in-water sensors that we use while we're out there. These measure things like temperature, salinity, but also the color of the water and how much light is being backscattered out of the water. This is an instrument that we have installed on Lake Okeechobee to hopefully look at these microcystis blooms. It's called a sea prism. This looks at the light that is backscattered off the surface of the water. And obviously, you can see the discoloration of the water from the microcystis is going to cause a lot of green light to be measured as scattering off the water and being measured by this instrument. And hopefully that'll give us an early warning of blooms happening on the lake and potentially blooms that may get transported to the St. Lucie Estuary. And then finally on the right here, this is an example of some really high resolution satellite imagery where you can really see a lot of these different colored water masses. And the color of the water here in these images is related to a lot of things, but primarily one of the important things that controls the color is the algae that's in it. And so we can use this to try and figure out where this algae is and where it's going. And finally, one of the cool things that I'm involved in is development of an in situ microscope, which is hopefully something we'll get installed in the lagoon or in the coastal ocean sometime soon. This is still being developed here at Harbor Branch, and we'll be able to deploy this instrument and it'll be able to monitor independently and visualize organisms like these types of algae. And hopefully we'll be able to see harmful algae from this data as well at some point. So I guess I'm getting close to the end here. I didn't realize how late it was getting. I guess I should leave some time for questions. So I'll end it there and acknowledge all the other people that are involved in this work, and I will take any questions.
Thank you so much for that fantastic presentation. We really appreciate your participating and presenting to the Water Ambassador Program. So take this opportunity to answer your questions or type your questions into the chat box, and Vincent and I will relay them. And then we will also be popping up the evaluation poll for you guys.
Hey, Malcolm, I just wanted to add to the whole birds tie-in with pseudonychian domoic acid. There is a study that followed up, and I tried to post it in the chat, but it wouldn't let me, where they looked at bird corpses from around that time when Alfred Hitchcock observed erratic birds flying around, and they did confirm that in their, I think it was in their guts with the food, the fish they were eating did have detectable pseudonychia cells in them.
Wow, that's fascinating. That's great. Oh,
Yeah.
I think you're getting off easy today. No questions, which just means that you're really comprehensive.
good Lord.
Mr. Osborne, does your class have any questions?
Oh, yes. Am I coming through?
Yep.
Terrific. Good. That mute button is hiding from me. Yeah. Sure. I have a question. I don't know to what extent your research includes historical data, but can you comment on, have these harmful algal blooms intensified as the population has increased, or is that beyond kind of what you've looked into?
So that's a really good question. I have not personally worked with a whole lot of historical data yet. And I've only been around here since 2015. I moved down. So most of my observations are more recent. But I think it's safe to say that in the Indian River Lagoon, particularly and locally across the Florida Peninsula, these harmful algal bloom problems are getting much worse. From everything I've heard, the water clarity in the lagoon used to be much better historically than it is now, and that's there used to be far more extensive seagrass beds, and now we see really turbid water quality pretty much year round in lots of parts of the lagoon, and significantly fewer seagrass beds around. And that's probably not all entirely due to algae. It may be other things, but algae is definitely a big part of that, especially that Oreoumbra lagunensis bloom, that persistent bloom that's up in the northern IRL. That started, I believe that was first noticed around 2013 or 2014, maybe even a little bit earlier. And it was previously unheard of. But since then it's stuck around and been causing a lot of problems, especially for the seagrass up there. And we know that inputs like nutrients, nitrogen, and phosphorus are not going down into the Indian River Lagoon. They're pretty high, and so there's no reason to think that this problem is going to get any better. And it's probably just going to get worse until we get a handle on some of these factors that are driving the blooms, like nutrient inputs.
Thank you. Very interesting.
Thank you. And I think we have time for one more question if anyone has one. And if not, I want to thank you again, Malcolm, for being a great presenter for the Water Ambassador Program. I want to thank all of our water ambassadors. Oh, we have another question. Okay, sorry.
I think we do have one more if you got a minute.
Yeah,
absolutely.
Hi. So I'm really impressed by all of the technology that you've shown that shows you that where the algae blooms are and and all that. So I was wondering how are you going to use that technology and utilize it to help stop the harmful algae blooms?
That is the $1 million question. And if I could answer that, then I'd probably be rich and famous. So the basic idea is that if we can figure out how these different environmental factors that we can measure, things like nitrogen concentrations, temperature, the amount of light they're getting, if we can figure out how those things are impacting the total amount of algae that we're seeing in the water and what kinds of algae, then we'll be better able to identify what steps we should take to mitigate these blooms. And we don't know the answers to all those questions yet. This is something that we still have a lot of work to do to figure this out. It's a very complicated problem. Like many biological problems are, all these organisms and environmental factors are interconnected in complicated ways. So it's really challenging, but it's really interesting at the same time.
Yeah, I assume the issue is like trying to figure out a way to stop the algae blooms without impacting the other aspects of the environment.
Yeah, exactly. You have to always remember that this is a natural living ecosystem. You know, there's a plan right now to treat the water that's coming from the lake with basically with hydrogen peroxide, I think, to kill the microcystis. Well, that's all well and good, but you know, that's basically going to kill lots of other stuff too. And like I said, these algae form the base of the food chain in these ecosystems, and we just can't kill them all. Otherwise, you end up with a completely dead ecosystem, and I don't think that's better than that's not a good solution to the problem, as far as I'm concerned. So it is very tricky.
Thank you.
But yeah, no problem.
All right. Any last questions?
Hey, real quick, Malcolm, I'm sorry, I don't want to take too much time, but I just learned about this morning. Can you comment on, is there any potential toxic effects of pyridinium cysts that might be in the sediment?
That's a good question. That's a really good question. My guess is that the cysts are not going to be as much of a problem for toxicity as the vegetative or the the other life history stages, the motile stages. But at the same time, there are organisms that live in and feed within the sediments, and those are in turn eaten by bottom feeding fish and things like that. So there is a potential pathway there for those toxins to get into the food chain. We have not done an extensive study of the cysts, but we know they must be there. And I believe other people have looked and found them, but that is something that should probably be looked at in much closer detail. The cells that we've found are always quite toxic. So I would expect there to be at least some toxins left in the cysts. And it could get into the food web.
Thanks, Malcolm. The question came up in the context of clam restoration.
Well, clams are going to be filter feeding the motile stages that are swimming around in the water column, not necessarily the cysts, and could potentially concentrate a lot of that toxin within their guts. So that's clams are a real important vector for getting these toxins into food chains, because they filter feed those organisms and they end up concentrating it within their bodies. So they can become extremely toxic, and that can be very dangerous if you go to eat the clams.
Okay, and Virginia just made a comment saying that she heard a theory years ago that clams stopped settling after a big pyro bloom year. So interesting. Well, it is after 1 o'clock. We promised you all that this would end at 1. So I want to thank you all for your time and for participating and for being water ambassadors for clean water in the Treasure Coast of Martin County. And thank you again, Malcolm, for your great presentation. And we look forward to seeing you all on our next webinar, which I believe is February 23rd, for the Indian River Lagoon report card. So thank you, and have a great week, everyone.
Thanks, everybody.
Thanks for having me.